Konnichiwa guys, welcome to our channel Loops of Biology. We will be taking today the next part of biomolecules, that is nothing but DNA. Alongside, we will be talking about what is the structure of nucleotide and what are the different names of this particular nucleotide. So coming to your first part, that is how the nucleotide structure actually looks like. So if we are talking about nucleotide, so in case of nucleotide, there was a presence of ribose sugar, there was a presence of nitrogenous base, and there is going to be the presence of phosphate group. So as I said that nitrogenous base is attached on first prime carbon of the sugar and phosphate group is attached on 5 prime carbon of the sugar. So if we are talking about a sugar, this sugar structure will look like this. Wherever on first prime carbon there is going to be the presence of nitrogenous base. As the structure extends, there is presence of hydroxyl group, and on 5 prime carbon atom there is going to be O, P, double bond O, OH, OH. So this kind of structure is basically called as nucleotide. Now what are the common names? What are common names of nucleotide based on nitrogenous base? So based on attachment of nitrogenous base, as the nitrogenous base is the only component which is continuously changing inside a particular structure. So what are the common names? So first one, if I'm talking about nucleoside containing adenine as your nitrogenous base, that is known as adenosine. Adenosine. This is the name of nucleoside. So this is nothing but my nucleoside, and this nucleoside doesn't contain your phosphate group. So whenever I am adding one phosphate, this name only changes to adenosine monophosphate. It's as simple as that. So there is adenosine as your nucleoside. So the nucleotide name will be adenosine monophosphate. If you are talking about guanine, so there will be guanosine, and similarly the same case for nucleotide, that is guanosine monophosphate. That with respect to thymine, so that is thymidine. Alright, the thymidine is a name of nucleoside, so there will be thymidine monophosphate. This all nucleosides and nucleotide are basically present with respect to ribose sugar. Whenever I am going to add deoxyribose sugar, every name will come with a prefix of deoxy. Alright, next case will be of your cytosine, that is known as cytidine. So there will be cytidine monophosphate, and coming to your last nitrogenous base, that is uracil, that is called as uridine, and the name will be uridine monophosphate. So these are your common names for nucleotides. Now coming to the structure of this nitrogenous bases and their numbering is quite interesting. So we have to see what is the numbering present inside your nitrogenous base and why is that important. Whenever I am talking about purine attached on a ribose sugar, then it is attaching from ninth position. So ninth nitrogen of purine is attached to 1 prime carbon of your ribose sugar. While if I am considering pyrimidine, then there is going to be first nitrogen group is attached on first prime carbon of your ribose sugar. So in this particular case, can you distinguish about purines and pyrimidine? What were they? Whenever I am considering purine, there has to be two rings. So guanine and adenine were your purine. So this is my purine, and similarly with your guanine, while other ones, all these three are nothing but not the three, but this cytosine and uracil are basically your pyrimidine. And there is one more pyrimidine, that is nothing but your thymine. So thymine is also belonging to the same category. Did you get this? So if I am talking about a structure of thymine, that is one of the important questions which can be asked as per your statement questions or assertion and reason questions are considered in the examination of NEET. So if I am talking about structure of thymine, it is resembling to uracil. It is resembling to uracil. As the uracil is having presence of ketone group on fourth and second position, as the uracil group is going to be having a presence of ketone group on fourth and second position, the thymine is called as 5 prime. It is called as 5 prime or 5 methyl uracil. So the structure is basically the same. So the structure will be like this. There will be presence of NH. There is presence of NH. And there is double bond O, double bond O. There is double bond. And on the fifth side, there is a presence of one methyl group. This is the structure of my thymine nitrogenous base that belongs to a category of pyramid. Right. So moving to your main part of this video, that is nothing but your case of DNA. Yeah, I know the NCERT doesn't cover DNA and RNA inside the biomolecule itself, but we have to study this particular part of DNA and RNA in detail, because in the next year, that is in 12th, in molecular biology, you will be studying all this property together. So it will just, we can say, drop down your efforts, which will you have to take in case of 12th. So we have to focus DNA and RNA in very much detail. So if I'm talking about DNA, the full form is deoxyribonucleic acid. Deoxyribonucleic acid. And as the name suggests, there is going to be a presence of deoxyribose sugar. Deoxyribose sugar. That's why the nucleotides, those are nothing but my monomers inside a case of DNA, are also deoxynucleotides. So there is, everything is nothing but deoxy in case of DNA. And this DNA is a stable nucleic acid. Where will you find this particular DNA? If I'm talking about prokaryotic cell, then in that case you will be finding this DNA in your cytoplasm itself, and the region is called as nucleoid. Right? While in case of eukaryotic cell, there will be membrane bound organelle, and that membrane bound organelle is nothing but your nucleus. And I hope everyone is remembering that there is discontinuous double membrane structure. So this nucleus will be having nuclear envelope which is discontinuous in nature. So now in this particular image on the right hand side, it has been seen I will climb up this strand of DNA to see where life takes me. Now why is that so? This man is saying, whenever we are talking about the complete length of DNA, alright, whenever I'm talking about complete length of DNA, this DNA comprise of your deoxynucleotides. And this nucleotide set is basically forming genes. And these are very important whenever any particular character is expressed within our body. Whichever character is expressed, whether it will be our skin tone, whether it will be our hair color, whether it will be our eyeball color, everything is governed by this particular genes. And these genes are nothing but a set of deoxynucleotides present in the complete length of DNA. So during a process of evolution, this length of DNA must have undergone mutations. Mutations are nothing but some changes inside the structure of DNA or some changes taking place inside the set of nucleic acid arrangement. So these mutations must have caused the changes in arrangement, right? And that arrangement must have adapted inside the genes. So these genes are finally making a structure called as protein. This is also one biomolecule, which is the important one. And these proteins are finally deciding the characters of organism. How organism is going to behave, how organism is going to look in its phenotype, that completely decided by the protein formed by genes which are made up of deoxynucleotide and present on the DNA. So complete life is governed by these particular proteins which are coming out from a code of DNA. So DNA is like a computer code which have to be decoded by something which are finally forming your proteins. So the complete code of life, nothing but your code of life is secretly placed, is secretly placed in a structure called DNA. So this is nothing but the introduction of DNA. So moving to your actually what DNA contains and which nitrogenous bases are there inside your DNA. Let's see. So if we are talking about your DNA, the discovery of DNA was done by your Watson and Crick. These were the two scientists who actually discovered the structure of DNA in 1953. The DNA might be in different structures. It can be your circular or it can be your linear structure. So if I am talking about prokaryotic cell, then there is your circular double-stranded naked DNA. While if I'm talking about eukaryotes, there is going to be your linear double-stranded DNA. Alright, so this DS is nothing but your double-stranded DNA. So in this particular structure, Watson and Crick were the one who actually discovered that the monomers of the structures are deoxynucleotides and the nitrogenous bases and the nitrogenous bases present inside this structure are nothing but adenine, thymine, cytosine and guanine. So ATGC, these are the four specifically nitrogenous bases present inside only DNA. These are specifically these four nitrogenous bases of DNA. They are not present in the case of RNA. So basically, adenine, cytosine, guanine are common, but the thymine is here specifically for DNA. This is present specifically in DNA. Now, what is this particular structure? The structure is here double-stranded helical structure. So the Watson and Crick have discovered this particular structure as double-stranded and it is spirally coiled. So it is nothing but your helical structure. And this DNA was commonly your bDNA, which is generally found inside organism. There are also various categories of DNA, that is aDNA and zDNA. So if we are further approaching and going to pursue biology as your career, then you must be going through this particular types of DNA ahead. So if I'm talking about your double-stranded helical structure, then there has to be some polarity. There has to be some polarity to strands. Now, how this particular strands are running inside the DNA? So if we moved back, there are two strands in helical pattern and each pattern has their own polarity. Now, what do you mean by polarity? Whenever I'm talking that in the case of chemistry, those are talking about the charges, that is positive and negative charge. But whenever I'm coming that inside a DNA, it represents on which side of nucleotide the particular functional group is free. Okay, so this is slightly confusing. So let's talk it about polarity through images. So there is a presence of nucleotide. Suppose this is my 5' end. And 5' end has a phosphate which is not bound to any of the functional group. And there is this kind of structure, P double bond O, OH, O, C, and this is my another set. So this OH is present on 3' end while the phosphate is present on 5' end, right? So whenever the 5' carbon has free phosphate group, so whenever the 5' carbon has free phosphate group, this end is called as 5' end of the DNA. Got it? And whenever there is presence of free hydroxyl group, free OH group on 3' carbon, that is known as 3' end. So the polarity of this particular DNA is starting from 5' to 3'. Alright. So one strand of DNA has polarity from 5' to 3', while the second strand of DNA will have polarity in reverse direction, that is from 3' to 5'. Now, why is that so? So this basically the both strands are anti-parallel are anti-parallel and complementary. So what this particular statement means? First of all, their polarity is completely different. So whenever I am reversing this particular polarity, the strands are becoming anti-parallel. Why can't we keep these strands parallel as such? So as the nucleotide suggests, there is going to be the presence of nitrogenous base on first prime carbon. So if I'm talking about parallel cases, then there will be, suppose this is my strand of DNA with a nitrogenous base with this kind of structure. This is what your phosphodiester bond is. So whenever I'm considering this as my one strand, which is having a polarity from 5' to 3'. So what if I kept same strand in a parallel case? Same strands in a parallel case. So there will be your phosphate group, there will be oxygen, phosphorus, double bond O, there will be your case of next set of sugar, and finally there will be OH. And again, the nitrogenous bases are exposed on the right side. Correct? So we have to form hydrogen bonds. We have to form hydrogen bonds between nitrogenous bases. So will it be possible if nitrogenous bases are far apart from each other? Right, it's not at all possible. So whenever the nitrogenous bases in in parallel arrangement of strands, in parallel arrangement of strands are exposed far away from each other, are exposed far away from each other. So they are unable to form hydrogen bond. And if the hydrogen bonds are not present inside a structure, then both strands cannot stay together. They cannot stick together. All right. So for hydrogen bond to form, the nitrogenous bases needs to come close to each other, needs to come close to each other. And that is only possible whenever, and this is possible when the strands are antiparallel. The strands are antiparallel. Now, one part of this category is explained. So, if I am talking about antiparallel case, that is completely clear. I hope you have got this particular point. If not, please comment down your queries in the comment box. Next case is complementary. Now, even if I am keeping the particular strands antiparallel, they have to complementary to each other. Now, what is complementary pairing? So, there is a case called as complementary base pairing. Complementary base pairing in DNA. So there is complementary base pairing. What does that mean? That is, there has to be specifically the bonding between purine and pyrimidine. So purine and pyrimidines are going to form bonds. So what are they? So whenever I am considering in the case of DNA, that is adenine bonded with thymine through two hydrogen bonds. Please be specific about the type of bond. There is going to be the presence of hydrogen bond between adenine and thymine, and those number are basically 2. While in case of cytosine and guanine, there is nothing but here 3 hydrogen bonds. So there are total 3 hydrogen bonds in case of cytosine and guanine, while 2 in case of adenine and thymine. I hope this is clear. So whenever I am anti-paralleling this particular strand, so even though the strands are anti-parallel, the nitrogenous basis has to be complementary, to each other. So they have to be complementary to each other. So this is one of the important case due to which the two strands of DNA remains attached to each other and stable. Remains attached to each other. And stable. So this stability is particularly gained by one case, that is deoxyribose sugar. Next case by complementary stranding. Third case, arrangement of strands in antiparallel order. And fourth case is nothing but your case of phosphodiester bond. Got this point? So if I am talking about a complete structure of DNA, these sugars are basically a backbone of your structure. So the deoxyribose sugar the deoxyribose sugar is acting as backbone of structure. Backbone of structure. And those are stacked on each other. And those are completely stacked on each other. Now, whenever I'm talking about backbone of the structure, that backbone should provide stability. So this backbone should provide stability to the structure. Now this stability is in two different forms. Your first case will be formation of phosphodiester bond, formation of phosphodiester bond, and next case will be site of attachment for phosphate group and nitrogenous bases. Correct. So there has to be site of attachment for phosphate group and nitrogenous bases, which is basically very important for the formation of phosphodiester bond ahead and complementary base pairing respectively. So if we are talking about the next part of this complete structure of DNA, this DNA is basically been seen by next scientist called as Erwin Chargaff. So one scientist was basically Erwin Chargaff, who has played a role inside the structure of DNA, deciding the number of purine and pyrimidine inside the complete DNA. All right, he has decided the total numbers of purines and pyrimidines in DNA. But this DNA has to follow double-stranded structure. Otherwise, Erwin Chargaff rule is completely flawed. So Erwin Chargaff, which is giving you your Chargaff rule, is basically saying that in case of double-stranded DNA structure, that is, in case of double-stranded DNA structure, the purine is to pyrimidine ratio is always going to be 1. So what does this mean? If I'm talking about purine, so that is A plus G, right? And pyrimidine is nothing but T plus C. That has to be 1. So this is nothing but A plus G combo is always equal to T plus C. So the number of purines and pyrimidine in case of the number of purine and pyrimidines sum is completely equal and having a ratio of 1. That is what Erwin Chargaff was saying all about your double-stranded DNA. Now, if I have to check, there are some organisms which are stable at higher heat, So how will their DNA will stand in that particular higher temperature? All right. In that particular higher temperature, the DNA has to dismantle. It has to completely broken down. But in that particular case of organism, that DNA is still stable. How is that possible? Because the DNA of those organisms contains higher GC content. Now, what do you mean by higher GC content? Higher GC content means number of number of cytosine and guanine pair are basically very much higher, and this as such is called as base pair. Alright, so adenine bonds with thymine is a one base pair. One base pair contains two nucleotides. Got this point, guys? So in this particular organisms will have higher GC content, and this GC content increases is basically increase in the number of hydrogen bond, and as the number of hydrogen bond increases, GC content increases. That means hydrogen bonds are basically increase, and more, as the number of hydrogen bonds are increased, then the stability is basically increase, because it will require higher heat or temperature to break this particular DNA. Got this point? Is there any doubt? All right. So now if we are talking about your GC content and AT content, but as G triple bond C is basically having 3 hydrogen bonds, so it is more stable, and A double bond T is basically having 2 hydrogen bonds, so they are basically slightly less stable than that of your GC. So whenever I am deciding the function of DNA with respect to surrounding, alright, whenever I am considering DNA function and structure, with respect to micro environment and surroundings. So what do you mean by microenvironment? That is nothing but what are the conditions present inside a cell, and surrounding means what are the conditions present inside actual environment. So as per depending on microenvironment and surroundings, the structure shows mutation. Alright, so the structure and function of DNA is completely based on microenvironment and surrounding. So it is regulating the structure of DNA, but that is only possible till the complete development. It's not like that if we are moving to some of the high temperature rich condition, then our GC content will automatically increase. It's not like that. So till the complete development, this particular DNA might get changed based on the mutation taking place inside it, completely dependent on microenvironment and the surrounding. Got my point? So now whenever I am talking about GC, G triple bond C, and A double bond T, both are called as base pairs, and it is represented by BP. So both are called as base pairs. So this base pairs, that is one base pair contain how many nucleotide, guys? One G and one C. So there has to be your C. So there is a case of GC. So one base pair contains two nucleotides. This relation is one of the important relation which can be required to solve some of the questions. So next case will be of dimensions of DNA. So this is basically main structure of DNA, how actually the strand of DNA look like. So this is the structure of DNA. Now as I am talking about the structure, this structure is completely based on X-ray diffraction pattern. So this is found out from X-ray diffraction pattern, and this pattern was found by two scientists, Rosalind Franklin, a lady, and Morris Wilkins and Morris Wilkins. Based on their data, the structure of DNA has come out to be your double helical structure. Thus, particular Watson and Crick have taken out the data. They just compared that with the sample they got and finally led to conclusion that the DNA is basically a helical structure with two strands. So if I'm talking about one strand with 5' end, that will end finally at 3', and the other strand as 3' that will end on the 5' side. So there is a polarity from 5' to 3' and 3' to 5'. So coming to the dimensions of DNA. So these dimensions are specifically about bDNA, the commonly found DNA. Alright, so this is your commonly found DNA. So first dimension is nothing but your diameter of DNA. So there will be diameter of double-stranded DNA. So what do you mean by diameter? It is nothing but distance between. It is distance between two strands. Alright, there will be a distance between two strands and that is 20 angstrom. So whenever I'm talking about a diameter, that means the distance between two strands, and this is basically your 20 angstrom, which is nothing but your 2 nanometer. So 20 angstrom is nothing but 2 nanometer. I hope angstrom unit is very clear. So 1 angstrom unit is 10 raised to minus 10 meter. 1 nanometer is 10 raised to minus 9 meter, and 1 micrometer is 10 raised to minus 6 meter. We have to remember this particular conversions because they are very useful whenever I am talking about dimensions of DNA. Next case is basically a length of 1 ton. A length of 1 ton. So if I am talking about length of 1 ton, then there will be starting from this particular point, we will be ending till this case. Alright, so this is nothing but my length of 1 ton, which is 34 angstrom unit. So there is going to be a length of 1 ton is 34 angstrom units, which is equal to your 3.4 nanometer. Whichever conversion you want to remember, that is dependent on you. So third case, how many base pairs? Okay. So number of base pairs present in 1 ton. So how many number of base pairs are present inside 1 ton? There are total 10 base pairs. So 10 base pairs are nothing but 20 nucleotides. Got my point? So if I'm talking about distance between, now that you have to calculate distance between two successive base pair. Distance between two successive base pair, then what will it be? So this is basically if I'm talking about this two successive base pairs. Alright, or this two successive base pairs. So what is the distance between two successive base pairs? As we have seen that there are how many base pairs in 1 ton? There are total 10 base pairs, and these 10 base pairs are arranged completely in the length of 34 angstroms. Alright, so this is basically arranged inside 34 angstrom for 10 base pairs. So distance between two successive base pair will be 3.4 angstrom, or it will be 0.34 nanometer. Got my point? Next case will be of the angle made by 1 ton. The angle made by 1 ton. The complete angle made by 1 ton. Angle of pitch. Angle of pitch. So this is nothing but your 360 degree. So the complete rotation of this particular angle of the strand of DNA, that is in 360 degree, while the angle between two successive base pairs. Can you guess it, guys? Two successive base pair. So we have to divide a complete circle with 360 degree into 10 parts. All right. So whenever I'm talking about angle between two successive base pair, that will be 360 degree upon 10. So that will be your 36 degree. So 36 degree is the angle present between two successive base pair. This is one of the important data whenever you are going for any dimensions of DNA or solving the questions of DNA. Got my point, guys? So coming to your first question, if the DNA has 20% adenine, if the DNA has 20% adenine, then what is the percentage of other nitrogenous bases? So we know that adenine is 20% of the total DNA. So if I am talking about the complete nitrogenous bases, those are 100%. So minus 20% is already there. That is 80% of the nitrogenous bases are still remaining. So we know that adenine bonds with thymine. So however, how many number are going to be there of adenine? The same number has to be there of thymine, right? So if adenine is 20%, then thymine has to be 20% itself. Got my point? Because adenine has to finally bind with thymine. So there should not be any thymine left without binding. So whatever the number of adenine are present, they has to be same as per your thymine. So out of 100%, the 40% is gone now. So remaining what? There is going to be cytosine is going to bond with your guanine. So there is your X plus X because their number is going to be same and remaining one is 60%. So there is X is equal to 30%. So adenine whenever there is 20%, the thymine is 20% again and cytosine is 30% and guanine is going to be here 30%. This will be my answer for this question. Next case, if the double-stranded DNA, if the double-stranded DNA, if the double-stranded DNA has 2000 nucleotides. Now they are talking about nucleotide. There is no base pair as such. Then what is the number of phosphodiester bonds in complete structure of DNA? Now they are asking for number of phosphodiester bond. Now the phosphodiester bond, first of all, phosphodiester bond is present between nucleotides. It is present between nucleotides. So whenever I'm talking about double-stranded DNA, whenever I'm talking about double-stranded DNA, there has to be total 4 polarities, right? 5 prime to 3 prime is one and 3 prime to 5 prime is another polarity. So there has to be your 4 groups, that is phosphate, hydroxyl, hydroxyl and phosphate group has to be free. So they are not forming any kind of phosphodiester bond, right? So they have given us with nucleotide number as 2000, right? So out of this 2000, if we are eliminating 4 of this particular nucleotide, then the answer will be 1996. So this will be your number of phosphodiester bonds. It's as simple as that. Whenever I'm talking about 2000 nucleotides and 4 groups are free, so they are not forming any phosphodiester bond ahead, right? So there is no phosphodiester bond, no phosphodiester bond. Phosphodiester bonds are formed on ends of DNA structure, ends of DNA structure, and as it is a double-stranded DNA, as it is your double-stranded DNA, there are total 4 ends. So that's why the total number of phosphodiester bond are nothing but 2000 minus 4, that is 1996. This will be my answer. Next question, it is slightly trickier. We will take some different calculation for this question. In the place of 10.8, we'll take 10. Let's see what is the question is saying. If I'm talking about if the double-stranded DNA is 10.2 kilo base pair long. Alright, let's write it down. The length of DNA is 10.2 into 10 raised to 3 base pair long. Alright, so the length is 102 multiplied by 10 raised to 2 base pairs. Alright, what will be the length of DNA in micrometer? If we have been given this particular case, in base pairs, then we know that one turn of DNA contains how many base pair? 10 base pairs. So there need to be X turns of DNA has 10200 base pairs. Got it? By simple unitary method, you can see that X is equal to 10200 upon 10, that is 1020 turns. So there are basically 1020 turns present inside a complete structure of DNA. Alright, so they have been asking us about the length in micrometer. Now we know that one turn is equal to 34 angstrom unit. Alright, that is nothing but your 34 angstrom unit. And that is basically, if we are talking about 1020 turns, then there will be 1020 multiplied by 34 angstrom. So this will be my length of DNA. But that will come out to be 1020 multiplied by 34 will be 34680 multiplied by 10 raised to minus 10 meter. Right? They are asking us the complete length in micrometer. So we have to shift 4 digits or the 4 decimals inside this particular number. So 1, 2, 3 and 4. So there will be 3.468 multiplied by 10 raised to minus 6 meter. That is nothing but answer will be 3.468 micrometer. This will be my answer in micrometer for the length of DNA containing 10.2 kilo base pairs. Got it? So if we are talking about your next part of the question, that is if the DNA has 50% of GC content. If the DNA has 50% of GC content, that means out of your 10 to 00 base pairs, 50% are basically of GC. 50% are of GC. So can you calculate this 50%? 50% is dividing by 2. So there has to be total base pair of guanine and cytosine will be your 5100 base pairs. Alright. So there is guanine and cytosine are basically having 5100 base pairs. And the remaining one that is A double bond T are 5100. All right, they are completely making 10200 base pairs. Now, whenever I am considering guanine and cytosine, we know that there are three hydrogen bonds. So multiply by 3. So the answer will be 00350. All right. And whenever I am considering adenine and thymine, there is going to be two hydrogen bonds. So answer will be 00210. So the total hydrogen bonds are basically 15300 plus 10200. So the answer will be 00552. So these are 25,500. So there are 25,500 hydrogen bonds. So the answer for next part, that is number of hydrogen bond, is nothing but 25,500 hydrogen bonds are present in the complete structure of DNA. I hope you understood this particular type of question, and this is one of the difficult question which can be asked on the topic of DNA. This is the complete DNA, guys, along with their general properties and the dimensions and the questions we have covered. I hope you love this particular part and you will be sharing this particular part among your friends. So please do not forget to like, share and subscribe to our channel, guys. Bye bye. See you in the next video.